Welcome everyone to this early career workshop in pulmonary omics, sponsored by the ATS section on genetics and genomics and the assembly on allergy, immunology, and inflammation. So these workshops are really designed to complement other resources that already exist for postdocs and junior faculty researchers. So it's not just early career advice, but it's really specific to respiratory omics research. So today's session is going to be titled Establishing a Research Niche in Respiratory Omics. We're very honored to have two really outstanding panelists here, Dr. Rachel Kelly and Dr. Russ Bowler, who are going to guide our discussion in how to establish your niche in pulmonary omics. So let's start with some introductions. So Dr. Kelly, would you introduce yourself?
Thanks, Matt. Hi, I'm Rachel Kelly. I am an assistant professor of medicine and an associate epidemiologist at the Channing Division of Network Medicine, which is based at Brigham and Women's Hospital and Harvard Medical School in Boston. My primary work is in metabolomics, specifically looking at early life asthma in children. And I'm also interested in integrative omics and trying to see what we can do by using more of a systems biology approach, integrating multiple different omic data types.
Great. And Dr. Bowler.
Thank you for inviting me, Matt, and others on the call. I'm Russ Bowler. I'm the chair of genomic medicine at Cleveland Clinic. I'm an MD, PhD. My MD training was in pulmonary and critical care medicine, which I still practice. My research expertise is in proteomics and metabolomics, particularly in chronic obstructive pulmonary disease, or COPD.
Great. Thank you both for being here. My name is Matt Moll. I'm a pulmonologist and junior faculty member at Brigham and Women's Hospital. And I'd like to invite my two co-facilitators to introduce themselves too. So David Zhang and Kaity Allen. So David, if you want to introduce yourself.
Thank you, Matt, and thank you for inviting me as well. My name is David Zhang. I'm a junior faculty member at Columbia. I'm clinically trained in pulmonary allergy and critical care. And my research interest is the genetics of pulmonary fibrosis, but also with integrating transcriptomics, especially single-cell transcriptomics.
And Kaity.
Hi, I'm Kaity Allen, and I am a director of scientific research for the Thomas Lab in the host microbe interactions department at St. Jude Children's Research Hospital. My research focus has been in influenza and host immune factors that are responsible for increasing severity of influenza infection, as well as genetic mechanisms of regulation of those host immune factors.
Right. Before we begin, I just want to point out that the main discussions of the workshop are going to be recorded, made available on the ATS website, as well as the Red Journal AJRCMB Lung Science podcast. There will be breakout sessions. Those will not be recorded, just for confidentiality reasons. And so just to kind of review the structure, we're going to have a quick roundtable discussion led by Kaity, and then we'll have our breakout sessions, and then we're going to reconvene and share our discussions with our panelists and get feedback from them and some guidance. So Kaity, if you want to take away with the roundtable discussion.
Yeah. So to dive into our roundtable discussion, we're going to be addressing 3 key questions with our panelists, alternating which of the panelists is going to respond first. And so we're going to spend about 5 minutes on each question. The first is, how and when does a trainee approach their PI about establishing a niche or branching out from the lab? So Dr. Kelly, would you start us up?
Thanks, Kaity. So I guess in terms of when, I suppose I would say it's never too early to start thinking about it. And I suppose with that in mind, the one thing that you really want to have is a good and kind of open relationship with your PI. I know that's not always the easiest thing to do, but I think, attempting to maintain that or start that communication from the very beginning means I think you don't necessarily have to worry too much about that exact time when you need to approach them, because this is something that the two of you will work on together and build together as you move forward. But I think in terms of how, the best thing you can do is really just read as much as you can, If you are able, attend conferences, attend talks, attend meetings, and try and work out what the kind of remaining questions are in your general field. I think with establishing your own research niche, you don't want to move too far away from your training, from the work your PI does. Obviously you need to be distinct, but I don't think you want to go so far away unless you have other support and means to do that. But I think just really working out what those remaining questions are, what those gaps are. Are there certain populations that haven't been looked at within your specific research question? Is there some controversy in the field that requires additional questions? So I would, be thinking about that again, as I said, kind of as early as you possibly can from when you start your training, and just maintain that open dialogue with your PI. Because they'll be the one, them and other mentors that you have will be the ones that can help a little bit more with timing. But I would say, I guess, never too early to start thinking about it. And, I'm speaking very much from a research perspective. I don't know if Russ has additional thoughts from a clinical perspective, which might be somewhat different. I'm not a clinician, but that would be my general advice.
What do you think, Russ?
Yeah, thank you, Rachel. That was excellent. I agree with everything she said. And I'd add too that it is the job of a mentor to teach a mentee on how to approach and start independence from the mentor. So it's really a two-way street. I think it is important, as Rachel pointed out, to establish what are the questions that haven't been answered that are worth answering. And also, as Rachel pointed out, don't stray too far from your skill set until you really have the tools to do that.
Those are really good points. Just from my perspective, I have a fantastic mentor, and he started our conversations off really early and really helped to guide me in the area that we study to make sure that I saw where the lack of knowledge was. So I appreciated the early conversations.
I'd like to probably, also add, because I didn't fully answer Rachel's question, that being a clinician is really not different from being a researcher in this regard, and that the clinician should think early on about what directions and maybe diseases or therapies that he or she might be interested in. But the principle is no different for clinicians as it is for researchers.
And I guess like Kaity, I'm very lucky that I've always had wonderful mentors. I don't know if Russ or Kaity or others, Matt or David, have thoughts on if you feel like you can't have the open and honest discussion with your mentor for whatever reason it might be. Does anyone have any advice for how to approach that situation? Because I've been lucky.
One of the lessons I've had from my amazing mentor is he always told me, he's like, I shouldn't be your only mentor, and you really need to reach out to other people because they provide unique perspectives. And I reached out to other people despite the fact that I felt very lucky with my own. And that was really useful. And so I think having the extra support, especially if you don't have a great relationship with your mentor, is really useful.
I'll very quickly echo what Kaity said. I think, having an advisory committee that is not directly involved in your research can provide a nice objective perspective on things, and not to mention it's a good thing to include, especially if you're going to apply for a mentor grant to have that training as well.
So for those of you who are clinicians, do you tend to have do you have clinical mentors and research mentors, or do you feel like it's better to have somebody who is a clinician researcher that can kind of address both gaps?
I think the ideal mentor is someone who is somewhat similar to you I'll give an example for myself. I tend to seek out clinician scientists as mentors, and I also point out that mentorship really never ends. Some people are surprised when I tell them, even as a chair, I have mentors that I talk to frequently for career advice. So it doesn't end, but I think it's easier to relate to someone who's has a similar area, either a clinician or clinician scientists or pure research scientists. Those are often people who can relate to you. And I would certainly relate best to those mentors, but it doesn't mean, as Katie pointed out, you can't have multiple mentors. Multiple mentors are in committees, are something we strive to provide for all our junior faculty, because it's a diverse perspective that's important.
It's been fantastic. I'm just for time's sake going to go to the next question. How do you maintain the agreement with your PIs so that it remains mutually beneficial after transitioning into transitioning to independence? Dr. Bowler, let's start with you this time.
Yes, that's something that every mentee or trainee needs to deal with. And the key is the independence part. So you need to find something that you can make your own. And a good mentor actually will allow you to do that, and then still be collaborative. I have lots of examples in my career of mentees or mentors that have allowed that independence. And the practical things you might want to consider, because this is evaluated when we look at grant proposals, for instance, is independent authorship. So it doesn't look good as if you're trying to be independent if your senior mentor is on all your papers. So I would encourage you to have those conversations early with your mentor about publications that maybe where it's okay to not list your mentor on the publication. And then have those discussions. Really, it's communication. That's the important thing. When you branch off, have frank discussions with your mentor about what you will take and what, maybe acknowledge what your mentor has done and will do.
Yeah, I would totally agree with that. I just agree with everything Russ just said. I think similar to the first answer, really keeping the dialogue open and being honest with each other is the best way to go about it and make sure things are mutually beneficial. I think to follow up on Russ's point about authorship, because again, this is something, that we're told over and over again, but it isn't necessarily always easy to do, because as a junior person, often the funding is coming from that mentor. So to negotiate not having them on the paper can be a little like politically tricky. So I think in that instance, and again, this goes back to that first question of establishing your own research niche, to the extent that you have the time and are able, and, always, of course, work with your mentor to agree in advance. See if you can find other groups who you can start to publish with, just to, get away a little bit from that idea of your mentor always being the senior author in your paper. So, especially, as we said, consortiums, the type of networks that you're building up as you think about your research niche, look to ways to see if you can publish with those groups as well. In terms of remaining mutually beneficial with your current mentor, I would say, be realistic about what your role is. I think, we all are kind of striving towards that independence, but I think, it takes a while to get there. So I think you do have to be realistic about what you are bringing to a project. And, if this is something that really is the paper that you need to be last author on, what is your role in that particular project? And don't try and jump the gun and, try and reach that independence and that last authorship position before you've actually got there, because, that's a surefire way to annoy the other senior people you're working with. But I think as well, once you are independent and once that's been established and, then maybe the authorship issue is no longer as relevant because you already have the promotion or whatever it is, keep collaborating with your mentor, keep working with them to, give back what they hopefully got to get you where you are, because even once you have your own niche, as we said, again, when talking about that first question, it's likely that your skill sets are still very relevant to each other. You can still work synergistically. So assuming that you have that good relationship, then, keep in touch and keep working with your mentor because, once you've established a good relationship, it's always nice to keep working with those people.
I'd add too that Rachel mentioned timing. Timing is very important. I wouldn't raise this subject with your mentor right upon starting. You have to put in the time, and you also need to communicate that why you're doing this with your mentor. So for instance, authorship. One of the purposes of not having a mentor on your manuscripts is to help other people see that you are independent. So have that discussion. I think many mentors will understand that, but timing is important. It's not appropriate to do that when you're in your first year of working with your mentor who's fully supporting you.
Those are all really valuable.
Oh, sorry.
Sorry.
I was just going to chime in and say that one thing that has helped me with senior authorship has been the projects that are unfunded projects. So sometimes you get master students or undergrads who want to do something, or residents and fellows. And if you can mentor them and teach them the skills, especially if it's like a project that's unfunded and not of high interest, those are the ones that I find most comfortable saying, I'll pretty much do this with this person, and then it would be okay if I was the senior author. And they're kind of like, this isn't even really part of my research, so thanks for helping this person because I'd love to help more people, right? So that seems to be a good formula for me sometimes. So
Well, that's very useful. And it's also really useful for the people who you're supporting, Okay, last question of the roundtable discussion. So in omics, we're often tied to specific data sets, but may need to move to a new geographic location. How do you detach from a specific data set? What do you think, Rachel?
Yeah, I think, it comes back to the previous points we've raised. You really want to, as much as you can within the confines of what you need to be doing for your training, where your funding is coming from your training, try and build relationships with other PIs who have other data sets. And again, a really great way to do that is through consortiums. I'm sure many people here are involved with TopMed and other consortiums like that. There's always those are a great way to both build a relationship and to access new data. Similarly, I think more than ever, there's a lot of publicly available data sets available. Leverage those as much as you can. And I think, when working with a specific data set, work out, when you're still in your training stage, work out what your transferable skills are, so that what you're learning is not so specific to that data set that it can be easy. I'm sure everybody does, anyone who's doing coding, that is a transferable skill in and of itself, but, focus on those transferable skills so that you can easily switch to another data set. And then I guess finally, just, as I said before, maintaining those relationships wherever you did train, so that depending on the rules and regulations around that data set, I think these days maybe there's less need to detach from a specific data set than there used to be. Again, assuming you can kind of find your niche within that data set.
Yeah, this is becoming a more common issue, and unfortunately, academic medicine is a little bit like a feudal system where your university or institution really sort of owns everything. So, and also controls access to the large public data sets that Rachel may be talking about. For instance, UK Biobank, TopMed, MVP, et cetera. And in all of those instances, you need to have your institution sign off that they're basically supervising your use of these data sets. So when you move an institution, you essentially detached from your previous institution and have to reattach yourself to these public data sets through the appropriate institutional channels. Don't forget to do that. That's becoming more and more important as as institutions become the gatekeepers for these data sets. For non-public data sets, this can be really challenging because, as I mentioned, usually your institution owns that data set, even if it's something that you may have had independent funding for. So I would encourage you to have discussions or when you're thinking about moving, have discussions with your mentor in particular about future access if you want to access that same data set and it is not public.
Russ, can I ask, because
Yeah.
oh, sorry.
Go ahead.
I was going to say, I don't really work that much with publicly available data sets. You know, that's such a good point. And I know that there's been some emails going around the chatting about the cost of the UK Biobank going up. I mean, do you think are some of these publicly available data sets becoming kind of cost prohibitive, particularly for more junior people?
I think that is a problem. I mean, I don't know if prohibitive is the right word for the UK Biobank data.
Yes,
And
I'm not
so
trying
you
to
say those, I'm actually, when I refer to public data sets, include those too, because those really, they're not public in the sense that anybody can access them, but they're public in the sense that you can apply for permission to access them. And in most of the time, they're sort of obligated to give that permission if you go through the correct channels. I think that is an issue. That's something we'll have to deal with. I mean, information is power, and it's really unfortunate. I think that certain groups like the UK Biobank will be charging for their data because that does inhibit research, and it really inhibits I think in the poorer regions of the world that don't have $10,000 or $20,000 to pay for access. And I know a lot of these organizations have special arrangements for underdeveloped countries that can't afford it, but I think it does put a little chill on research to charge for access to data.
I'll add a quick, so maybe a different perspective as well. I think for the non-public data sets, if you have the tools or the knowledge to create another data set in your new institution, for instance, your different geographic institution, mirroring the way that the data set was collected in your original institution, that would also be a very rich opportunity to, follow the same protocol and the same pipeline for success from before, and then you've automatically created now a replication data set that'll allow you to continue to work with your mentor. So, there's a lot of good examples of this where in my field in ILD, where Justin Oldham, for example, went to UC Irvine and then went to or UC Davis rather, and went to Michigan, and now he has 2 replication data sets. So I think if that is something that probably not an option for everybody, but if that is something that can be done, it can be another way to take advantage of a situation like that.
Great. Well, thank you. So that was great. We'll move on to the breakout sessions now. So they're going to be 3 groups. We're just going to do 10 minutes, and you can go ahead and choose your groups. So there will be, I'm going to put it in the chat right now for everyone. So here are the 3 groups and kind of things to discuss. So one question is, how could you find data that would allow you to establish your niche after defining it a priori? How do you develop transferable skill set that is unique from mentors, apart from simply finding a new omics data type? And then how do you transition from mentee to mentor, especially in the context of omics? So can we open up the breakout rooms for everyone and they can choose? All right, see you in there, okay? All right, welcome back. So let's go through each of the groups and we'll just take a few minutes to kind of discuss. So for group 1, would the spokesperson just be willing to summarize quickly what you all talked about?
Sure. So in our talk, we mentioned about first will be challenging in certain kind of disease, for example, pediatric disease, pediatric ARDS, or something like IPF, because the number of sample size very small. So it's really need to get a network in order to get a data set. So this is the first challenge. The second challenge is for some data, some omic data, especially downstream from the genomic, for example, the metabolomic, proteomic, because they use a different kind of platform. So it's hard to harmonize the whole data set in order to do a larger cohort for the analysis. So this is the second challenge. The third challenge is we talk about, a lot of kind of sometimes have some administration barrier, like even like UK Biobank or something, if those institutions haven't worked with this data set in the past. And then we also talk about where to find the data, and we talk about multiple kind of data repository source we can use. And then finally, we got some tip from Dr. Russ Bowler, talk about how are we able to get the data from individual researcher, why he recommends we can approach as a collaboration way instead of so they will be more willing to share, especially with the new data set they put up ever on. If you approach it as collaboration, then they're more easy to willing to share. This is our summary.
Okay, awesome. Thank you. Dr. Kelly, maybe you could give some feedback on what you just heard.
Yeah, I really like the idea of just approaching everything as a collaboration. Because I think, if you're lucky enough to have a good mentor, that probably means that they're exceptionally collaborative. They're very generous with, the data that the questions they give you to work on, the data they give you access to, the positions on the papers that, they put you. So I think just maintaining that in your mind as you move forward in your career, the more senior you get, just remember that that's how we all move forward, right? Team science, being collaborative, and that's by far the best way to get people to be open and willing to work with you and share your data, and I guess more importantly to work with you again after the first time. So yeah, I would just totally agree with Russ's advice on that one.
Great. And Russ, do you have any other thoughts related to this one, or?
The other maybe addition would be search repositories, and as I mentioned, the comments, which I don't know if we'll hear more about. Rachel is a great example of a consortium that could allow you to find new data for metabolomics. But there are other sources like NCBI Geo for transcriptomic data, dbGaP for genetic data. So learn how to use some of those data sets. And then, yeah, the other approach is really just learning which cohorts have which data, which is sometimes could be a challenge, but just reaching out to people that you might meet in conferences or in talks.
Okay.
Yeah, I don't know if anyone is working in metabolomics and is not familiar with COMETs, then in the interest of time, I won't go into it now, but do feel free to contact me because there's a huge wealth of data there that we would, we just want people to work on. So yeah, if you're interested in that, please do get interested in metabolomics. Please do get in touch. I can give you more information.
I was talking you up and selling you in group one. So group one knows all about it.
And the lung working group is ongoing, Russ. I am still working the COMET analytics team. It's been quite the saga, but we are almost there.
Okay. All right, great. So group two, maybe your spokesperson could summarize quickly.
Okay, I will summarize very quickly. We had talked about essentially when you're in a training period, you are developing a lot of skills that makes you an expert compared to your mentor who's, maybe a few steps back from the data. And so there's a lot of different ways to look at data. There's a lot of different types of analyses that can be used on the same data set. There's a lot of different ways to become an expert in these omics protocols. And now there are so many different single cell protocols is my example because it's the one that I use, where, changing to a different kit, or changing the way you process samples, or what my lab calls hacking protocols, really allows you to step into a different, project type and question just by making that one little change that makes your project different from the rest of the lab. And so taking those data sets, learning how to analyze them a bit differently, taking those protocols, learning how to process things differently, allows you to have a skill set that's distinct enough from your mentor's expertise and allows you to have that transferable skill set.
Awesome. Dr. Bowler, what feedback do you have for the group or advice?
I would say similar feedback. As was mentioned, as a trainee in omics, you are probably going to be even on the cutting edge and maybe more advanced than your mentor. So I don't think it's usually a problem to transfer those skill sets.
Okay. And Dr. Kelly, any other?
Yeah,
thoughts about
I was in that group, and yeah, we were just kind of laughing about how, and now how our mentees have got so many more skills than we ever did when they know all the, and I think the example in the question, said other than switching to a different omic, and I think, we talked about actually switching to a different omic is probably the absolute hardest way to build your niche, and probably in most omics somewhat unnecessary, because as Katie said, we talked about single cell, but in the context of metabolomics, you've also got things like spatial metabolomics, there are new developments within an omic happening all the time. So I guess I would say, unless you want to, and of course that's also absolutely fine, I think I wouldn't recommend switching omic. I would just recommend, looking at what's new within the omic that you're already working on.
That's great. That's really interesting to hear. And group 3, Eric is our spokesperson for group 3.
Yeah, so our question was how to transition from mentee to mentor, especially in the context of omics. And I guess we had 3 kind of things that we discussed. The first was, I think Matt had talked about this at the beginning, but just as you transition to that mentorship role, be very open to sort of taking on trainees at all levels. And it's fine and kind of fun to take on undergrads and maybe med student, that type of thing. And it doesn't have to be, some massive, nature medicine project. The second thing that we talked about was one of the biggest changes from early in a K to kind of getting that first R is just there's a massive explosion of responsibilities. Like you just get pummeled with department requirements, division requirements, society requirements, journal requirements, etc. And so unless you're intentional about being a mentor, that's the one thing that can get lost in the sauce, because it's like one of those things that's the most important thing, but low urgency. And so unless you make it urgent and carve out time in your schedule, it's so easy to get lost because you have research coordinate, all this other stuff that sort of are urgent, but not important, and it can get lost. And then the third thing that we talked about was kind of what Katie was saying earlier is, as a trainee, you are going to have a niche in terms of a technical skill set that is highly valuable to other folks, clinicians, etc. And so as you transition to your primary project, still maintaining and sort of leveraging that expertise. And so maintaining yourself as a middle, kind of helping on aspects of different projects to build new collaborations that you can sort of mature into sort of independent projects. So always having your 1 or 2 primary projects, but really leveraging your expertise to sort of collaborate on 3 or 4 things that you have in your back pocket that you can kind of spring forth from. I guess those were kind of the 3 things that we discussed.
Yeah. All right. Great. And so Dr. Kelly, any thoughts about this?
No, I think that was perfect. That's exactly all the things I would have said. I think one thing, just to, I guess, another shout out for ATS, is that, in addition to mentors within your institution, there are opportunities, and I can't remember the exact details now, maybe we can share them after if someone can remember, but I know that within ATS, and whatever other organizations you might be part of, I know that's something we're looking at in Comets. You can also kind of seek out mentoring opportunities there. So, I guess just thinking wide, when thinking about, where to look for mentees. And then I guess, to go back to Eric's point about being conscious about mentoring, I agree, it's so hard. I think especially when you're kind of in that middle stage of your career, because there are so many things you need to be doing. So I think that advice of being conscious about being a mentor and mentoring opportunities is a really, really fantastic advice.
Great. And Dr. Bowler, anything?
Yeah, just quickly, when you're approaching that time where you're thinking about transitioning to become a mentor, many institutions have training on how to be a mentor that's really targeting first-time mentors or early mentors. Take advantage of those opportunities and take them seriously because they will provide the tools for you to become a good mentor.
Okay. Yeah, I didn't know. I actually wasn't aware, and I'm sure there are many at my institution, so that I should take advantage of. Great. Well, anyway, so I wish we had more time. There are definitely more things to discuss, but I wanted to thank everyone for coming to the workshop today. We've discussed some strategies for establishing your niche in pulmonary omics, finding data, developing transferable skills, transitioning to a mentorship role. Obviously, a special thanks to our panelists, Dr. Rachel Kelly and Dr. Russ Bowler, for their guidance today, and my co-facilitators, David Zahn and Kaity Allen. Also want to thank the ATS staff for organizing this, Tara and Samantha. This should be posted on the ATS website and the Reg Journal Lung Science podcast. If you have suggestions for future workshops, definitely reach out to our section, and I'm happy to do these things. If you want to be involved, definitely shoot me an email, and happy to include more people. So thanks again.
Thanks,
Thank you
everyone.
very much, Matt, David, and
Thank you.
Katie. Bye-bye.
Thank you, everyone.
Bye.